Okay, on deck is oral presentation number 20. This is San Marino, California, Team 2. This is a Mission 6 finalist proposal, just selected for flight and going through flight safety review.
Hi, my name is Sarah Higdon. This is Charmaine Sue, Lauren Takayama, and Kiri Yamamoto, and we are the San Marino, California Team 2. And our proposal is titled Cycloastragenol in Microgravity, Its Effects on Telomerase Activation and Telomere Longevity in Saccharomyces cerevisiae, the instant strain of Baker's yeast. Next slide, please. Next slide, again. Thank you. So a telomere is the end sequence of chromosomal DNA, which effectively protects the coding DNA during the replication process. Each time a cell divides, the DNA divides along with it, and each time the DNA divides, the telomeres get shorter and shorter until they are non-existent, in which time the coding DNA is lost and the cell dies. Telomerase is an enzyme that rebuilds these telomeres by adding sequences on top of the already existing ones, promoting cell growth. There are many new drugs that claim to activate telomerase and effectively euthanize tissues by promoting local cell growth. Next slide, please. So our experiment was formulated to qualify whether or not the most potent of these drugs, cycloastragenol, does its job, as well as its effects on telomerase and telomere longevity in microgravity. The cycloastragenol anti-aging drug was chosen to be the best stimulator out of two other candidates, TA65 and astragaloside 4. We chose our drug because of its price, potency, and status as a newer drug. We also chose instant yeast because between human and yeast cells, the yeast cells have significant benefits, such as the fact that they do not need to be tested by NASA for disease, they have less complicated DNA, and their responses are comparable to human cells.
Next slide, please. Other materials that we utilized in our experiment, in addition to the yeast and the cycloastragenol, included bacto yeast extract, bactotryptone pancreatic digestive casein, D-glucose anhydrous, and C-plaque GTG for an agar medium, phosphate buffer saline, and agarose. Next slide, please. Like majority of the other groups, we had planned to present our ground truth experiment today. We initially wanted to run a ground truth experiment using fluorescent in situ hybridization, acronym FISH, the protocol that we would have used to compare telomere lengths between our ground and space experiments had our proposal been selected. However, after a long discussion with our sponsor at City of Hope, we came to the conclusion that it was simply too expensive and resource consuming. We were advised that it would be wiser to continue fundraising this year. In lieu of the possibility that we improve our proposal and try again next year, we have enough funds to overcome the costly difficulties that come with using FISH, including cell wall lysis of our yeast cells, determining yeast telomere sequences, and developing telomere primers. With this consideration in mind, we figured it would be better to perform our ground truth experiment at a more opportune time. However, since we didn't want to pass up this great opportunity and certainly didn't want to come empty-handed, we performed two preliminary experiments instead. Next slide, please. These experiments were necessary to determine whether, number one, our yeast cells would actually grow at all in microgravity, or in our case, simulated microgravity, and number two, whether our drug, whose effects have only been tested so far in humans, would negatively impact or elicit abnormal responses in organisms with cell walls, such as our yeast.
Next slide, please. To test if yeast can grow under microgravity conditions, we simulated microgravity using a C-plaque agarose. If the yeast can survive while immobilized and limited to using surrounding nutrients when in the agarose, then the yeast cells would be able to grow in suspension or a microgravity condition. When we used two mixtures for our experiment, an independent mixture of YPD and yeast, and a dependent mixture of YPD, yeast, and C-plaque agarose. Both mixtures were used and went under the same procedures, and the results were analyzed using a 10-fold serodilation method. The two yeast concentrations of the mixtures were about the same, thus proving that the yeast growth was not affected by the agarose. Next slide, please. The top left picture depicts two mixtures with yeast added. The top right picture depicts the yeast growth. The bottom left picture is after the yeast growth, and after the two mixtures were put into the centrifuge. And our last picture is of our two diluted solutions.
Next slide, please. To determine whether cycloastragenol negatively affects yeast growth, we created two mixtures, one containing cycloastragenol, yeast medium, and yeast, and the other containing only the yeast medium and yeast. Since most of the cycloastragenol studies have only been performed on human cells, we wanted to see whether cells with differing cell structures, such as cell walls, would react differently. So to create an ideal environment for the cells to grow, we placed them in a lab line orbit environ shaker for an hour and a half at 30 degrees. We then centrifuged the mixtures and removed the excess yeast medium. Then we used we also used the 10-fold serial dilution again, taking some of the original mixtures and adding buffer, and taking some of that new mixture and adding more buffer. We then placed the new diluted solutions in a spectrometer, which measures the concentration, and we found that the concentrations of both solutions were marginally different. Next slide, please. In the top left picture, you can see our mixtures right after yeast has been added. The left picture. In the top right picture, you can see the mixtures after yeast growth and after they've been centrifuged. In the bottom left picture, you can see the two mixtures after the first dilution. And in the bottom right picture, you can see the two solutions after the second dilution, and in the cuvettes, which are ready to be placed in the spectrometer.
Next slide, please. After performing our experiments, we concluded that yeast can grow in simulated microgravity.
And cycloastragenol does not negatively affect yeast growth.
Both of these experiments were necessary to confirm that our proposal would be plausible and the results extrapolative to humans. Seeing that both outcomes of our preliminary experiments met our expectations, we venture to stand by our initial hypothesis that in the absence of gravity, cycloastragenol will still activate the telomerase, which in turn will likely restore telomeres at a faster rate than telomerase on Earth, resulting in longer telomeres and telomere longevity.
Next slide, please. Thank you.
I just want to say that I misspoke. This is a finalist proposal for Mission 6. And just like the North Charleston, South Carolina team that we heard yesterday, they reproposed and they got to the flight level after reproposing. I believe Wyeth Colo, your community program director, is looking at Mission 8. So I would urge you to explore reproposing this wonderful experiment. Are there any questions for the San Marino team?
Hi, so Tim from Goddard Space Flight Center. So I had never heard of cycloastragenol before. Are you telling me that my bread yeast and I are in imminent danger of living forever?
Well, there aren't many studies actually on cycloastragenol or any anti-aging drugs for that matter, especially on yeast. It's because it's a relatively new drug. We don't really know if anything can live forever. I don't know if that's possible yet, but that might be a danger that we should look into as we traverse the anti-aging world.
Okay, are there any more questions? No. Let's thank the San Marino, California team number 2.